Hello everyone, and thank you for attending today's webinar titled Resolving Single-Cell Heterogeneity Through Single-Cell Multiomic Analysis by Dr. Tyznik. Dr. Tyznik is our Associate Director of Immunology Applications, an experienced scientist and a great asset to BD. Before we begin, we wanted to cover a few housekeeping items. At the bottom of your screen are multiple application engagement tools you can use. All the engagement tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right corner. If you have any questions during the webcast, you can submit them through the Q&A engagement tool. We will try to answer these during the webcast, but if a fuller answer is needed or we run out of time, it will be answered later via email. Please note, we do capture all questions. We encourage you to download any resource under resource list that you may find useful. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. Webinars are bandwidth intensive, so closing any unnecessary browser tabs will help conserve your bandwidth. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your computer speakers or headsets are turned on and the volume is up so you can hear the presenters. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical questions located in the Help Engagement tool at the bottom of your screen. A recording of this broadcast will be made available after the presentation. At this point, I welcome Dr. Tyznik to please start his presentation. Thank you.
Thank you, Mahi, and thank you everyone for joining today. As Mahi mentioned, I am the leader of the immunology applications team at BD Biosciences. And today, we will be talking about resolving cell heterogeneity through single-cell multiomic analysis. So for those who aren't familiar with multiomic analysis, multiomics is essentially the generation of really biological data and analyses of multiple omes simultaneously, whether it's the genome, whether it's the transcriptome. And today, specifically, what we're going to be talking about is how you can leverage BD's technology to interrogate proteins on the cell surface, a part of the proteome. And along with that comes mRNA information that may be valuable on a single cell basis, part of the transcriptome. So as many of us that are on this webinar today are immunologists or are in the field of immunology, we probably have a significant appreciation for the value of single-cell multiomics. The ability to get complex and comprehensive data from single cells and also be able to apply that across a large population has been highlighted as the method of the year in 2019 from Nature Methods. And as we look in the literature and as we all move forward in the experiments that we're doing, we're all really becoming very much aware of how much information we can actually gain by integrating all of these technologies. No longer are we looking at what is the genome of a specific cell or maybe the mRNA transcript of those cells at one given moment, but instead we can answer questions like, what are the proteins that they're simultaneously expressing, or have they downregulated? How does that protein and mRNA correlate? And then what does that mean for our research or the patients or the disease that we're studying in their particular area of research? And of course, when you think about BD, many of us have traditionally thought of us as a company that provides antibodies and flow cytometers and cell sorters. But it's much more than that. In fact, it covers the entire single-cell analysis and discovery pipeline. all the way from the blood being acquired in, from the phlebotomist and the tubes that they're collected in, to the tissue dissociation using reagents like tumor and tissue dissociation reagents, to applying antibodies to define different cell populations within a heterogeneous cell population, to, of course, the sorting of cells or the analysis of them using a flow cytometer. However, as technologies have evolved and things have progressed, we've basically moved into a new space where we're able to scale proteomics even further. And along with that, drive discovery, which involves single-cell RNA sequencing as well as protein analysis simultaneously, which is a part of the single-cell RNA sequencing platform called the BD Rhapsody system, which we'll talk about that a little bit more as we go through this presentation. In addition, we often have the computational analysis component. Now, this is often challenging where people can generate a tremendous amount of data and information from platforms where you're doing single-cell RNA sequencing or proteomic and genomic analyses. And there's often sometimes a bottleneck when it comes to the bioinformatics analyses. And not that those specialists are no longer needed, because they certainly are, but we often find ourselves in a place where we want to just take a look at our data. We want to understand, do we want to move forward with the data that we acquired? Can we take an initial look at it and understand maybe what populations are present and do some exploratory and discovery ourselves? And that's where our informatics solution, which incorporates both Flojo and the single-cell analysis platform SeqGeek, which is integrated into Flojo, enables our researchers and individuals to take their sample all the way from a sample collection all the way to a high-level, system-level profiling using bioinformatics tools. So today, really what we're going to be talking about is how do we simplify the complexity? These experiments can become very complicated, or at least they may seem complicated. But once you understand what the workflow is and what it's like to go through this process, hopefully you can have confidence to be able to perform these types of experiments. Because when we think about, again, these end-to-end solutions, we cover blood and tissue collection. Everyone's very familiar with cell staining, how to basically stick an antibody onto a cell. Very familiar with those techniques, as well as flow cytometry, cell enrichment, and cell sorting. And the new area that I want to highlight to basically show how you can use these technologies, how they can integrate into your workflow, is the multiomics aspect, which is the BD Rhapsody system we'll be talking about, as well as our informatics for analysis for flow and RNA sequencing analysis. So when we think about when we want to do single-cell RNA sequencing, well, before we really had true single-cell RNA sequencing, I am an individual who has done this top line here, where we would do traditional RNA-seq. For example, I would look at a knockout mouse, and I would look at an infectious disease in that mouse and compare it to a wild-type animal. And then I would say, well, I want to look at the immune response in the spleen. And then maybe I was actually saying, well, I specifically want to look at a certain cell population called natural killer T cells. Well, sure, I could sort out the natural killer T cells, but, you know, in this initial approach, I would still have a very much of a bulk population, and there's a lot of heterogeneity within there. And even if I'm doing some pre-enrichment, the nuggets of gold, basically, these red cells, are probably the ones that I'm most interested in. But when I did this bulk RNA sequencing, essentially anything that was unique about these two red cells would just be lost by averaging of the signals. And we soon realized that we thought this technology would be leading to all of these amazing discoveries. And instead, it was just kind of ho-hum data. Sometimes we would get things interesting, but you know, it didn't give us the results that we were looking for. So what then happened is people would start saying, well, let's do single-cell RNA sequencing. Let's do serial dilution so that only a single cell would fall within a well, and then we're going to do sequencing of the RNA for all of these individual wells. Well, of course, you're limited by capacity. How many plates are you going to be able to do this with, and how many are you going to be able to sequence? But you also have the problem that the cells that you're looking at are still very rare. So you're spending a lot of money and resources getting single-cell information and data from cells that are kind of irrelevant to what it is that you're looking at. However, if you're able to directly sort your specific cells of interest directly into individual wells, you can focus what it is that you want to look at to your particular cells of interest. Now, traditionally, people would take these individual cells and look at ways to deposit them based off of protein expression on the cells to define the cell population or characteristics that they're interested in. Now, from here, you would get your mRNA information, and you could ask a lot of interesting questions from your experimental to your control. And if you have the capabilities of something called index sorting, you could correlate what was found in your mRNA with the proteins that were expressed on your cells based off of your flow cytometry panel. Now, that was well and good, but we're often limited by the number of proteins that we look at on the cell surface. And certainly, I know for myself, when I sorted cells, I wasn't interested in just adding more antibodies just because. I was just interested in adding what I needed to get the cells I needed into the individual wells. So your protein was very limited in terms of the information that you could get. So there's need for an improvement of technologies. And now you could ask, well, why do you need your protein expression? Well, if we take a look at what would be a perfect correlation between mRNA and protein abundance on the left, and if mRNA equals protein, we would get this nice linear line. Of course, that's not how biology works. Biology likes to throw all sorts of curveballs, and the reality of how mRNA correlates with protein is here on the right from this publication from Nature Genetics in 2013. And looking at these different log scales, you could see that unfortunately mRNA does not equal protein. In fact, it's far from it. And now, of course, I'm a biased cellular immunologist. To me, I think protein equals function. I use flow cytometer because I want to learn about the function of cells because I want to look at proteins. And that's why I look at BD Biosciences as really a protein analysis company. We enable the interrogation of cells for protein expression. And this correlation tells me that if I'm just focusing specifically on my mRNA expression and information, I'm probably losing a lot of information about what the cell is actually doing, because mRNA just tells you can make the protein, but it doesn't mean it's there. It doesn't mean it's functional. It doesn't mean that it's going to, if I have a CD8 T cell, that it's going to be actively attacking and killing something because it actually has granzyme A or interferon gamma. Instead, I might just be reading out it has the potential to do that. It may just have mRNA for those proteins, but no actual protein present. So that's where the BD-AppSeq and BD-Rhapsody single-cell analysis system really comes into play for the simultaneous protein and gene expression analysis. Now, I won't go into this technology too in-depth because on the BD Biosciences website, there's countless webinars out there that really highlight exactly how this technology works. But I'll give you the 50,000-foot view. Essentially, we are able to take all of the antibodies that we have that are normally conjugated with a fluorochrome and instead conjugate them with a unique oligo tag. For example, let's pretend this antibody that I'm on this slide is CD4. So we know that the epitope binding domain of this antibody will only bind CD4. Well, if it clone and it's a good antibody, which we've tested and validated though, so you can be comfortable that that's the case. But you don't know it's CD4. So, and if we don't have a fluorochrome on there to light up to tell us it's FTSE, because it would be FTSE CD4, what we've done instead is put an oligo tag on here, and there's a specific barcode or sequence that tells us this is a CD4. Now, it would be cute if the oligo sequence actually spelled out this is a CD4, but it is not. But instead, our bioinformatic pipeline can say that if I capture this antibody and this attached to this oligo, it was a CD4 antibody that must have been bound to a CD4 protein. Now, we have unique antibody-specific barcodes for our catalog of over 300 different antibodies currently available from BD Biosciences. So what you can do is you can take your cells and you can add your oligoconjugated antibodies to your cells. Now, how many different antibodies can you add? Can you add 10? Sure. Can you add 20? Yeah, absolutely. 40? That's doable. And we even have experiments and data showing all the way out to 80 or 100. We are still all learning how far we can push this technology, but essentially you can take all of these antibodies, stain your cells just like you stain any other proteins. If it was fluorochrome conjugated, well, it happens to be oligoconjugated, and label your cells. Now, using our Rhapsody system, again, we're able to float our cells across this basically honeycomb-looking cartridge where individual cells fall into individual wells. Now, once these cells have settled in there, each cell is now basically like a single-cell deposition if you're doing self-sorting. It's not exactly like that, but basically put them in there, but they do go in there. And now that cell is in isolation, and we can now add a bead. And this bead settles within this well and where the cell is located. And once we lyse our cell, we liberate all of those antibodies that were attached to the cell, as well as the mRNA. Now, as those are released within this particular well, they're captured by this bead. Now, each bead is uniquely labeled. There's millions of them. And they're basically uniquely labeled such that when we recover this bead later on, we know this bead came from one single cell, and all of the stuff that it captured came from that specific cell. So if there's 40,000 molecules of CD4, we'll catch 40,000 antibodies. Well, maybe it's not a direct correlation, but exactly 40,000, but it might be. It's pretty close. And so once we're able to capture this information, we can now send it downstream through a sequencing process, similar to what you would be doing with single-cell mRNA, but now you're getting all of this protein information simultaneously with all of this mRNA information. Genomics approach. So what does that data look like? You know how I mentioned we have ability to analyze our data in FlowGel? Well, on top is flow cytometry data looking at cells that were stained with fluorochrome conjugated antibodies, looking at primary markers, secondary markers, and tertiary markers. And we're familiar with this top line or top row of bivariate plots, right? Like this is what we expect to look at when we look at different cell populations. And I can look at my expression of CD45RA versus CD27 or CD38 information, or even a tertiary marker such as CD357. And I look at this data and I'm pretty happy with it, right? I can clearly resolve all the populations of what I'm interested in. However, if I take the exact same sample, send it through and stain the cells with AbSeq, and then go through the bioinformatics pipeline and get my sequencing data back, I then just take that data and import it directly into FlowGel. I can gate and analyze my cells the same way as if there's a fluorochrome conjugated antibody. And these are the exact same samples. And when I take a look at data and when I like to compare data, the first thing I ask are, am I able to resolve my populations? Basically a percentage check. Am I getting the same percentage of cells? And of course, I could go through and say, yes, I look at all of these and they look pretty equivalent. Although the more important things that I like to ask, the second thing is like, well, how good am I resolving my population? Just because I can see the population doesn't mean I can easily resolve it. And then if we look at CD45RA versus CD27, it looks like I can clearly resolve these populations. And even if I look at these secondary markers, which are even more unique, I could look at CD38 versus HLA-DR, and I certainly could resolve my CD38 positive, but I could also even say that I can clearly resolve these additional what looks like clusters or populations within that CD38. And of course, then CD357 looks comparable as well. The third thing that I like to look at when I'm evaluating my data is the spillover spreading error. What is the spreading of my negatives? So if I come over to my first plot where I'm looking at my primary marker and I look at CD45RA on the y-axis versus CD27 on the x-axis, if I look at the spread of my negatives, what does that mean? I'm looking at the sample that's CD45RA negative. How far does it spread up and towards my CD45RA positive? And if I look at my scale on my y-axis and I look at 0, and then I can see that my spread of my negatives go all the way up to 10 to the third, which is a significant amount of spread, all the way down to minus 10 to the third. And that's using my bi-exponential scaling so I can envision or visualize that entire CD45RA population. Now, I can tell you I didn't intentionally, or we didn't intentionally design this panel so that it would have that spread. This is actually a pretty good panel. I'm pretty happy with this resolution. But what really draws my eye about the AbSeq technology is that 0 is 0. If there is no counts, it is 0. Now, you could say, well, Aaron, look at that plot. It looks like it's kind of spread around in a circle or whatever, and it looks like it's going below 0. That's basically to build the density plot, to visualize it. They kind of have to build up the layers. Think of a topography map, and you're looking at it, the density and number of cells there. We have to have some layers in there. But if we remove that, I mean, all of these events are essentially at 0. We've removed that spillover and spreading error component, the thing that is very, very challenging when we're trying to build flow cytometry panels. And what does that mean? It means we can basically get really good resolution on the low end in high plexi panels. And so, of course, this is just examples of some of the data that we can get off of this particular platform. But I'm a little more interested in actually applying it to samples of interest. And that's what I'll be talking about next. So the samples that we've chosen in this particular experiment is just to apply this technology, both single cell, or flow cytometry cell sorting, and combining that along with the single cell multiomics platform followed by analyses. And so we're using chronic lymphocytic leukemia as our model or our system that we want to look at. And CLL is the most common form of leukemia in adults. It's characterized by a progressive accumulation of functionally incompetent lymphocytes in both the blood and bone marrow. And the standard for diagnostics is basically immunophenotyping. You can see that the CD19 positive cells are very clonal. And of course, some of the recent data that people have generated looking in CLL patients have identified that actually all CLLs are not created the same, and there's actually significant heterogeneity within these CLL samples. And so it really indicates that there's value in kind of evaluating the mRNA information as well as what is the protein profile to understand the unique differences in these CLL populations. And so here we're going to demonstrate the use of the BD Rhapsody single cell analysis system to simultaneously analyze protein and gene expression in single cells in clinical CLL samples. So what does that look like? Well, this is basically the experimental design and workflow. This is very similar to any other experiment that we would be doing, except we take it a little step or a couple steps further by doing single cell RNA sequencing on the cells. So we have 3 healthy samples, normal peripheral blood, and then 4 CLL samples, again, peripheral blood. Now, how do you stain these antibodies for AbSeq? Well, you basically stain them as if you stain any other fluorochrome conjugated antibody. In fact, in this specific experiment, what we've done is we've put unique sample tags, which basically defines each of these individual donors, so a total of 7 sample tags. We stain individual tubes first for each sample, and then we add all of the BD-ABSeq antibodies in this particular assay, which happens to be 32 antibodies, along with the antibodies that we need for our cell sorting, so 7 fluorescently labeled antibodies. So we can add all of these simultaneously to all of these individual tubes, and then the reason we multiplex or use the multiplexing kit is we can combine all of these samples together and then deconvolute them on the backside during the bioinformatics analysis. So once we've stained all of these cells, we'll go ahead and load them onto our cell sorter, sort out our CD19 positive B cells. Those cells we can then load onto our Rhapsody system and then capture the mRNA and protein that was released by the individual cells, perform cDNA synthesis on those beads, and those beads themselves can actually be stored until we're ready to proceed with library prep. Once we decide that we are ready to prepare our library prep, we have chosen to evaluate a targeted panel for the human immune response panel, along with a supplementary panel of additional genes we wanted to look at. Now, there's a lot of value in looking at targeted. Often we don't need a whole transcriptome analysis, and essentially we can really save some of our resources and even and time and money possibly by only evaluating the mRNA or transcripts that are of interest versus getting all of the information that may not be particularly relevant to the question that we're asking. So from here, following our library preps, we can move to our next generation sequencing, get our files back, and then analyze in the SeqGeek software. So we can look into this a little more granularly. Now, when we want to sort these cells, when it comes to these particular experiments, the more we can simplify them, the better off we're going to be in terms of workflow, concerns over possible errors, and also getting out very clean and interpretable data. And along those lines, you may have seen in the previous slide, we highlighted a FaxMelody sorter. Now, the FaxMelody cell sorter is basically a benchtop, easy-to-use cell sorter so that you can quickly set up your instrument and not need a tremendous amount of expertise to be able to isolate the populations that you're interested in looking at. And as a result, it fits in very well when we're trying to just get good basic enrichment of the cells that we are looking at for our single-cell multiomics analysis. So to ensure that we had simplicity in our design, our panel design involved making a lineage depletion, basically, style of panel where we used all of our lineage markers that we were not interested in on a common fluorochrome, that being APC. We then assigned CD19, which is our positive marker for the cell population that we're interested in, on a marker, or on a color PE. This allows us to make a very easy to stain panel, as well as minimal compensation due to spillover and spreading errors, as these fluorochromes don't really have much for interaction with each other. This allows us to quickly stain our samples and get a highly enriched CD19 positive population that we can then use for our downstream multiomics analysis. Now, we then have our 32-plex ABSeq panel that we also stained at the same time with markers that we were interested in looking at in our downstream data analysis. Now, what's a reason for cell sorting? Well, as you can imagine, we're interested in CLL and we want to evaluate the differences in these different CLL samples compared to healthy donors. And if we want to really kind of focus our time and money into the cells that we care about, it makes sense to just isolate out the cells that are of the highest value, which for us happen to be the CD19 positive cells. It allows us to exclude cells that aren't of interest and can also save time in terms of the number of cells that you need to load into your cartridge system, but it also allows you to effectively add more cells to your analysis. So CD19, of course, there's probably a lot of those cells that are present, but imagine if you're looking at something like a T follicular helper cell. It's a rather rare cell population that you're trying to look at. Now, if you wanted to look at 20,000 of those cells, you would have to do single cell analysis on hundreds of thousands of cells, basically, to try to find the TFH cells that are of interest. So we've chosen to just isolate and enrich for the CD19 positive cells. And now what we do know is that within CD19, we can use CD27 to subset different cell populations of the B cell component or compartment. Now we could have used the flow cytometry panels. As you look in this plot to the right, we could use the flow cytometry panel and sorted out all of these individual populations. But if we were to do that, we would basically have, depending on which populations you want, I mean, probably 3 different populations that we would be sorting times our 7 donors. And then, of course, then we are going to be doing downstream analysis on this. And instead of adding those markers at the flow cytometry stage, it's probably much easier just to throw in those 2 markers on the AB-seq side. So this data that we're looking at here is actually the data that came out from the 32-plex AB-seq panel, where we can gate on CD19 positive cells from our sorting, so all the cells are CD19 positive, and now we can just look at CD27 versus IgD. And it clearly defines our B cell subsets, and we can gate on these cell populations just like we would with flow cytometry data. It really lowers the learning curve to be able to analyze and interrogate our data. We now have a 32 marker panel here, right? Like we can gate on all of these different populations and start to evaluate the expression of CD117, CD33, and so on. Now, all of those are great, but of course, doing that approach, but we might as well take advantage of some of the new advanced algorithms that are available, the different plugins that are available in SeqGeek and FlowJo to actually do various principal component analysis and UMAP analysis if we want to use Monocle. And now I feel like I'm just saying the names of a bunch of things, but there's a tremendous number of actual algorithms that can really help you dig into your data, get more out of it because it's so rich. There's just so much information available. So as I mentioned, this upstream sorting really allows us to purify the B cells that we are interested in for downstream analysis. The BD Rhapsody single cell analysis system, the type of information that we can get out of that. So as we kind of think about our workflow, we really want to kind of focus and drill in on what we can get from the protein analysis as well as our mRNA, and allowing us to do this in a very simple and straightforward experimental design and workflow. So as you had seen previously, the BD Rhapsody scanner that is a part of the BD Rhapsody system is actually primarily used as a QC tool. So there's really 2 components. One component is the BD Rhapsody Express, and that allows you to load your cells onto the cartridge. And that requires nothing more than an electronic pipette that is a part of the purchase of the BD Rhapsody Express platform. Now the scanner is actually really a useful QC tool when it is needed. For example, you're able to use it to help guide you through the loading of your cartridge. It tells you the viability and concentration of the sorted cells, and you can basically use that to predict what is your recovery going to be on your sample. For example, if I go into the loading of my cartridge and it looks like I have decent viability, and by the time I start getting into my loading of my cartridge for mRNA analysis, the viability may have gone down significantly. And that's going to greatly impact the recovery of viable cells that come back through the bioinformatics pipeline. Keep in mind that when we think about these samples, I mean, the downstream sequencing costs can be high, and we like to have a point where we can say, yes, we want to move forward, or no, maybe we want to repeat the experiment or try to get better samples, and also allow us to troubleshoot if we have poor sample quality going in. Because unfortunately, mRNA is not the most stable once cells start to die, and that's an important QC metric of your data coming out of the bioinformatics pipeline. Of course, the Rhapsody system can let you know if there's the defined number of captured cells and how many cells are multiplets, as well as a number of cells that are loaded with beads. Now, it was noted in our specific experiment, one of our healthy samples had poor viability and low cell concentration and was excluded from the rest of the workflow. You may have noted, I did point it out earlier, we did start out with 4 healthy controls and 4 CLLs, but we now, moving forward, decided to focus on only our high viability good samples, which is now down to 3 healthy controls and 4 CLL samples. So as I mentioned, on the BD Biosciences webpage, if you go into resources and you can search for webinars, there's a lot of webinars that really go through the process of how all of this works. I know I'm just kind of glossing over it, but if you really are interested in the technology and the details, again, there's plenty of webinars and other presentations that go through that aspect of it. Of course, I think it's pretty cool, but unfortunately, I don't have time to discuss it here. But if you have questions at the end, we can also ask them. You can ask them at that time. So since we decide to capture our mRNA and our AB oligos on beads, we can then do a cDNA synthesis directly on those beads once we recover them. So basically, think about each bead now equals a cell. So we can store these beads, essentially the equivalent of the cell mRNA and protein, up to 12 weeks before we proceed forward with our library preparation and workflow. Now, a couple of things that I didn't focus on too much on the workflow is these types of things really allow you to kind of plan and stage your experiments so that, for example, sequencing can be done in one run with multiple samples, for example. It really minimizes batch effects. The same would be true if we're talking about trying to do sample multiplexing, where we can load all of our samples together. They're all mixed up. They're all being treated the same. They're all recovered the same. They're all going through the same library prep. It really can help you minimize any type of batch effects in between your experiments because, again, at the end of the day, we're looking to answer biological questions, not find limitations in technology. So in this particular experiment, though, with this flexible workflow, it really gave us the flexibility of waiting for more clinical research samples to arrive before we decided to do our full library prep and our sequencing. And we're allowing us to basically process our samples on different days as needed. So as I mentioned, we've now gone through and we've taken our 3 healthy samples and our 4 CLL samples. They've received their own unique sample multiplexing tag, which allows me to separate them on the back side, on the bioinformatics side. They've all been labeled with our 32 antibodies and our fluorescent antibodies. The CD19 positive cells have been sorted and loaded onto the Rhapsody cartridge such that a single cell has fallen into single wells. We've then captured the corresponding mRNA and protein information. Before we actually did our lysis and capture of the beads, we QC'd that process using the BD Rhapsody system. And now during our library prep, we focused on a BD Rhapsody targeted human immune response panel. This allows us to look at over 300 genes along with supplemental genes that are relevant to looking at human immune responses. We send this through our next generation sequencing and then import our data into our FlowJo software, and now we can really interrogate and ask interesting questions about the data. What are the differences between our CLL patients or our CLL samples and our healthy samples? So I think this slide really shows the power of single-cell multiomics. Now, we've all seen Tyznik plots before, and we know that it creates these interesting little islands, and it basically is taking all the information that it's receiving, and these algorithms are basically saying what's like what, or what's similar to other cells, for example. And on the far left, if we just use, and this is the same data that we have, with our 7 different samples that have been color-coded based off of the individual samples. If we take our RNA sequencing information only and then do a Tyznik visualization and looking at our individual populations and overlaying with our 7 different donors, you could say, yeah, I can see my individual donors or individual samples. They seem to be clustering nicely. Now, that's with RNA sequencing information only. In the center, we look at AbSeq information. Now, this is 32 antibodies, and 32 antibodies on the surface of these CD19 positive cells, and do a similar analysis. And as you can see, again, you can clearly start to define and identify your individual clusters that seem to be correlating very nicely to individual donors or individual samples. For example, I can start seeing that these cells in the AbSeq only in the upper left-hand corner look like a couple of CLL patients, and then the healthy seem to be located lower below, and they're all kind of clumped together. However, if I combined the information together of both RNA sequencing and AbSeq, now you can start really seeing the granularity and heterogeneity and distinction of these unique cell populations just by combining all of this bioinformatic information. Now we can then take these individuals or this analysis where we have RNA sequencing plus AbSeq, and we can begin to overlay and look for different protein expression. For example, in this, in the far left plot, you can see that we're just overlaying the CD5 oligoconjugated antibody expression across all of these donors. Now, if we look here, you can see that on the top area of this particular Tyznik map, where if you go back to our RNA-seq and AbSeq only, or RNA and AbSeq combined, corresponds to our CLL samples, that there's a significant amount of CD5 expression, which is what we would expect, right? Like that's a hallmark of CLL. And of course, then we can also look at a marker such as CD11C or CD10, as well as CD20. And this, based off of color, basically allows us to look at what is the level of expression that we're looking at across these different islands, and particularly these specific samples. So of course, as I was mentioning earlier, when we looked at our different clusters, we can see that actually our healthy samples generally cluster similarly. All right, so we can see that they're in this lower area. Of course, the way these Tyznik maps are generated, the closeness of one to another doesn't indicate necessarily absolute similarity or information being construed, but we can see that in general, the healthy samples, you know, there's different, you could say, clusters of which comprise all of the different donors, right? Like they're not all light blue and all dark green and all light green as individual islands. They almost kind of blur together, which would be representative of normal, healthy CD19 positive B cells. They would be just kind of all mixed together because there's nothing really particularly unique about any individual's CD19 or B cells. I mean, we like to think we're all unique, and maybe our cells are at some level, but for the most part, I mean, they're pretty darn similar. However, when we look at our CLL samples, I mean, they might as well be islands to themselves in the ocean, right? Like CLL patient or CLL sample 1, 2, 3, and 4 are very distinct, and they don't overlap with any other CLL, which is kind of interesting, which tells us that each one of these CLL samples in themselves are very unique, and that they're certainly not overlapping in any way with our actual healthy samples, telling us that there's a significant amount of heterogeneity even within these 4 different CLL samples. Now, of course, we could probably even zoom in a little further on these individual CLL samples because there's one that actually already draws my eye and maybe drawing your eye already as well. Now, of course, I put a little gate on these, or a little circle on these 2 populations in CLL4. And, you know, it's just our natural eye, you know, our eyes are very sensitive to patterns and then little changes. And, of course, when we take a look at these things, we might have already been drawn to say, huh, I mean, that looks like it's a little unique. You could say it's something unique about CLL number 1 as well. It looks like there's a little island as well, and there's even to 2. But right now, for this particular data discussion, we're just going to drill in onto sample CLL4 because it looks like there's some rather unique aspects to that particular cluster. So as you can imagine, so far, there is a tremendous amount of data that we generated, and I could spend hours and hours talking about it because there's, just from one single experiment, there's many, many different bits and pieces of information and potentially many answers that are hidden within there. So I could just even start out with like 3 rather basic questions here, right? Like, what drives the different clustering of the healthy and CLL samples? So why are they uniquely distinguished from each other? And then what drives the different clustering between the CLL samples themselves? They're all CLLs. I mean, wouldn't you think they would be similar in some way? And then taking it a step further, what drives the different clustering within the CLL sample number 4? So to do that, what we start out by doing is looking at all of our clusters from our Tyznik map here, which is on the right, which is the same one we've been looking at, which is healthy versus CLL samples, with CLL samples kind of focused more on top, and then our healthy samples focused more on the lower half. And now we can start overlaying some of these AB-seq reagents that we've been looking at. And now this can look like there's a lot going on, right? So obviously, to make it easy to orient people, if you go straight to the center where everything's red, they're all CD45 positive. All right, so of course they're all lymphocytes, and so everything's expressing CD45. But then when we start looking a little more in depth across the different markers and looking at the different expression, we can really kind of drill in on some differences between healthy versus CLL, then within the different CLL populations, and then, of course, what's unique about CLL4 in terms of that particular population looking almost like it's 2 independent clusters. So now if we want to really start looking at what drives the distinction and clustering most likely for these individual samples, we can start looking for differences between the healthy and the CLL. And I will draw your eye to actually CD279. Now, CD279 is the third row in the fourth plot over. Now, you can see CD279 that is now font color is actually, when we look at our CLL samples, all of our CLL samples are positive for having bound the CD279 antibody. Meanwhile, all of our healthy CD19 positive B cells are negative for CD279. Again, this type of information allows us to understand that that's part of probably the reason why these CLL samples are being clustered away from our healthy samples, but it also tells us a little bit about the nature of these CD19 positive cells from these CLL samples. Now, of course, we want to also understand what's driving the separation of the heterogeneity, for example, within the CLL samples themselves. And so I could then draw your eye to CD133, which is the second row down and the second plot to the right. And you can see that 2 of the CLL samples are positive for CD133 and 2 of them are not. And again, this is just a tremendous amount of information just by a quick glance at these protein expression overlays that's telling us something about what proteins are expressed in these specific CLL samples that's probably driving this different clustering that we're visualizing. And meanwhile, I can, from the same data, it's just astounding that I can start realizing, hey, I can even see differences for the CLL sample 4, where it looked like there's 2 separate clusters. So if I go to the second row down and the 7th plot over, I can look at CD45RA. And if I have an arrow pointing to CLL sample number 4, it looks like half of them are positive for CD45RA, or at least very bright, and the other half are like moderately bright. And then if I draw your eyes down 2 rows, if I look at IgD, it looks like there's also, with the arrow pointing to the population or the cluster, different levels of expression of IgD across this CLL sample. Again, a tremendous amount of information just by looking at different protein expression. And of course, I can take this even deeper by looking at a single-cell heat map. So if I want to look at a single-cell heat map where I'm just looking at combining both mRNA and proteins or AB oligos, I can ask the question, what makes healthy samples different from CLL? And all of these genes, this is just a representative of a handful of mRNA transcripts as well as some AB-seq antibodies that kind of distinguish the difference between CLL versus healthy. For example, healthy tend to be positive for CD81, CD20, and CD10, and CLL samples tend to be negative. I can then ask the question, what's the difference between the expression pattern of in CLL compared to healthy? And in this analysis, when I look at the mRNA genes or protein, you can see that, well, 2 of these samples are, CLL samples are very positive for CD5 antibody AB oligo expression, CCL2 and CCL1 to some level, as well as CD27 seems to be positive for our CLL samples compared to our healthy controls. Of course, then I can dig in even deeper and look at things that start making our CLL clusters rather unique and dig into what is up in our CLL samples compared to our healthy controls here, where we can see that, for example, the CLL samples, CLL sample 3 and 4 have a number of genes that they're positive for, which are not necessarily turned on in CLL 1 and 2, and vice versa. And what's really interesting, though, is if we take a look at something like CD69, I was talking about this earlier, where mRNA and protein are not always a good correlate. And if I take a look at my CD69 mRNA in my CLL sample 3, it's positive, right? Like I can see mRNA for CD69, and I can also see corresponding CD69 antibody expression, right? Now, if I then look at CLL sample 4, that's actually positive for CD69 antibody as well, but mRNA is incredibly low. And then I can look across CLL 1 and 2, and there is no CD69 antibody expression on those cells as well. Just driving home the fact that there's a lot of heterogeneity even within the CLL samples. And of course that is probably indicative, and it explains a lot why we have this unique clustering of these cell populations for each CLL, and really drives home the fact that these cells or these samples are in fact very, very unique, and reveals further that heterogeneity of CLL itself. And finally, I can drill in and just highlight our genes and proteins that are uniquely expressed on CLL4 compared to not being present on CLL1, 2, or 3, and also how that relates to healthy controls. For example, CLL4, as we saw earlier, is positive for or subset of the cells are positive for 45RA, IgD, CD117, as well as CD34. So again, this type of analysis through FlowJo and SeqGeek analysis really allows us to dig in and evaluate our data at just previously unappreciated levels. Combining both this mRNA and protein expression can help us really dissect the cell populations and understand what is possibly going on in these individual samples, to understand what is their protein profile and what is their mRNA profile. And of course, it's often a rabbit hole with this data. You know, I still want to better understand actually what's going on in the CLL4 sample. And so we can also then dig into that population a little more in depth. And so to do that, what we can do is we can analyze our data, as I mentioned in FlowJo, using AbSeq markers as if they were just a fluorochrome conjugated antibody, right? So taking a healthy donor, I can look at CD27 expression versus IgD, and healthy donor number 2 has a very nice separation and identification of the different B-cell subsets, switch, non-switch, and memory naive. Allows me to really confirm what I already know about the biology. And then I can apply that same gating criteria and strategy across all of my CLL samples, CLL 1, 2, 3. Of course, when we look at CLL4, now it's even more clear and distinct that there's something going on with this particular CLL sample. And if we then look at our corresponding AbSeq in this heat map as well, we can clearly see that they are CD45RA, and some are positive and some are IgD positive, and some of them are IgD negative. And that's probably one of the main distinctions between this particular sample with these potentially 2 clones that we're looking at in this particular CLL4 case here. And then, of course, they all seem to have a significant expression of CD27. So if we then want to ask the question of what's the difference between the IgD positive and the IgD negative populations, of course, we can then now look at differential gene expression in the heat map as well as protein expression on those 2 subsets. And as not surprising, when we just identify those cells that are IgD positive and look at differential gene and protein expression between IgD positive and negative cells, again, we can identify a number of both proteins as well as genes that uniquely define these 2 different populations within the CLL4 sample. So again, this allows us to really interrogate what is actually unique about this particular CLL sample. And then we're able to basically get or understand the heterogeneity within one specific CLL sample, how those, all four of those CLL samples compare to each other, and then how all of those CLL samples compare to a normal healthy donors or healthy samples. And of course, interestingly, we have confirmatory data from when these samples were collected and from some flow cytometry information. And even though we've used this AbSeq and single-cell multiomics approach to really interrogate and dig into these populations, we kind of knew from the beginning that there was going to probably be something unique from sample CLL4. Because as I mentioned, we have this original flow cytometry data looking at CD79B, which actually was not in our AbSeq panel compared to CD23. And in that scenario for CLL4, we also you're able to identify basically these two discrete populations within the CLL4 sample. And again, that's not surprising because as we know, the most common diagnostic approach for looking at CLL is using immunophenotyping, often through flow cytometry. Okay, so in summary, I hope you appreciate the value of the ease of these workflows when we kind of put them all together and going from your sample to answer, and how critical it is often to think about what are the populations that I'm interested in, and then enriching for those particular cells, because it's important to understand that you really want to get the most precise and cost-effective approach when you're performing these assays. And if you are interested in particular cell populations, you know, I would basically advise you to interrogate the ocean and instead look at the fish that you're actually interested in. So in summary as well, we've talked about the simultaneous analysis of protein and gene expression really enables deeper cell analysis, and that we observed a lot of the expected differences between healthy and CLL samples, and that this deeper analysis further revealed heterogeneity both inter and intra CLL samples. And ultimately, a complete workflow solution for enhanced single-cell analysis is a powerful tool for enhanced characterization of really any heterogeneous biological system. And with that, I would like to thank you for your attendance, as well as a thanks to the great team that I work with at BD Biosciences, who makes all of this possible, as well as the collaborators that we work with to help acquire our samples.
Thank you, Aaron, for the wonderful presentation.
Yes, thank you very much, Mahi. All right,
so I was just wondering, before we go ahead and open the forum for Q&A, I just want to make sure that folks can hear us clearly. If there's an issue, can you let me know? Okay, thank you. So before we open the forum, I'm really glad to see an engagement of this scale. We're really excited to see all the questions coming in. I wanted to take this opportunity to introduce Joseph Musmacher, who is our field application scientist who supports the SEM platform. Joseph has been in the field for over 20 years, and he has tremendous experience in the field of genomics, seeing how technologies have evolved and how BD is positioned, poised in a way to kind of combine many multimodal technologies to bring solutions to your lab. So thank you for joining us, Joe. And before the broader questions are kind of taken in, there is a consistent cadence of queries that came along, and I thought we could just answer them from the onset. There's a lot of questions regarding number of cells that can be loaded on the Rhapsody cartridge, cell viability, amount of cells that are harvested in, or the recovery rate, and the cost of the total experiment overall. So I know it's a bunch of questions lumped together, but Joseph or Aaron, if one of you could please answer them at the onset and we can engage.
Yeah, great. Thank you very much, Mahi. So as Mahi mentioned, we have an incredible expert on the call today regarding the field of genomics and single-cell multiomics. And so, Joe, if you wanted to kind of tackle a couple of the questions that had come in with regard to loading of cells and cell recovery on the Rhapsody platform, as well as roughly what it looks like when performing these experiments in terms of costs and so on.
Joe?
Thank you. I was on mute. Yeah, so the system allows us to query as little as a few hundred cells, up to as high as 20,000 cells per cartridge. If one was to take advantage of the multiplexing kit, we could permit people to go as high as 40,000 cells per assay. The workflow in general is quite simple. Obviously, there's a unique step to BD. The first portion of the assay is simply isolating single cells into one of our cartridges and harvesting the messenger RNA from the cells onto our magnetic barcoded beads. From that point on, there's a simple cDNA synthesis step, followed by a couple of PCR steps for enrichment, at which point people can proceed to Illumina sequencing. The assay generates an Illumina sequencing-ready library. Any other questions?
Yeah, thank you very much.
Yeah. Go ahead, Anne.
Yeah, I think another question that had been coming up was about cell recovery when we're using the platform. And of course, this may be sample dependent, but go ahead.
Sure. Obviously, there are a number of factors that can determine the ultimate capture rate, if you will, but there are a couple of statistics I typically use in terms of capturing cells in the microwells. We typically recover, or we typically capture upwards of 95% or more. But what I typically refer to is the stacked yield, meaning once the cells have gone through the entire assay in its entirety, what winds up being sequenced, officially we say greater than 65%, but what I see in practice is much more along the lines of 75 to 80%. And again, this is determined by a number of factors, for example, cell viability and cell size, for example.
Yes, thank you, Joe. Another question that we've been seeing quite a bit had to do with, we have targeted mRNA, which is what I talked about today, and then as well as cataloged available AbSeq, which is oligoconjugated antibodies that fit into this workflow. Can you speak a little bit about some of the alternatives? Because there were some questions about custom RNA or custom AbSeq or providing purified antibodies. So please share some of your thoughts on some of those offerings that are available.
Sure. So both on the gene panel front as well as the AbSeq front, we do offer, obviously, the off-the-shelf AbSeqs and the off-the-shelf gene panels. These are PCR primer panels. But we also offer an ability to generate entirely custom gene target panels for the PCR portion, up to about 500 sequences. Conversely, if you liked what was being addressed in the off-the-shelf panels, for which there were a couple more options, we can take the off-the-shelf panel and supplement them with up to another 100 gene targets as well. For the AbSeq portion, of course, we have our resume of AbSeq-ready proteins on our catalog, but one can also take any of BD's antibodies and convert that into the AbSeq as well through our custom processes. Likewise, if you have your own clone in your fridge or your freezer that you'd like to use it in the assay and convert to an AbSeq, we could do that as well.
Thanks, Joe. Another couple questions that I had been seeing had to do with the analysis, and maybe I can take this part right now. so there were some questions about do you need to have training in R or Python to be able to perform the analysis, and then how do you begin to analyze the data? And so first off, I'm an individual who started to learn R, and then I decided maybe I'm going to do something else with my time. Not that it is impossible, but I just didn't necessarily see the value for myself having that skill set at this particular moment. And so that's where the bioinformatics pipeline that BD offers, it's called 7 Bridges, that takes your sequencing data and provides all of the counts and information, the counts of mRNA, as well as AB-seq expression into a CSV file, basically, that can be imported into FlowGel. Now, if you've used FlowGel before, you're probably familiar with that interface of how you look at cells, how you gate things, and there's various plugins that you can use to do UMAP or Tyznik if you're doing high-dimensional protein analysis. Well, if you want to analyze your sequencing data, there's something called SeqGeek, S-E-Q-G-E-Q, and that is part of FlowGel. And it has the same feel and experience as what you're doing with your traditional flow cytometry data. However, it's actual sequencing data. And the data is basically the counts, which when we think about counts, we would traditionally refer to the MFI, which is a surrogate for the relative amount of antibody that had bound, which kind of corresponds to roughly the amount of protein that's on the surface of the cell. And that's your semi-quantitative way, basically. I shouldn't say that, but it's a really, it's not a precise measurement of the amount of protein, but it's a surrogate for it for us to look at the data. So when the data comes from the sequencing, which is the counts of oligos, and there's various controls that ensures that there's not PCR bias and so forth, that you basically get a raw read of number of oligos that were captured from AB-seq or mRNA transcripts. And so that information is just numbers and counts, and that goes right into FlowGel. And then once you get that information, you can just analyze your data in a similar way that you were analyzing with flow cytometry. Now, I had an interesting conversation with someone about a lot of the plugins and tools that you need to have some normalization of the data possibly for your analysis, or you might have to do downsampling or some sort of principal component analysis, things that allow you to kind of normalize and standardize the data while looking at it. And all of those are available in the SeqGeek software as part of your QC process in your analysis process. And of course, FlowGel has a tremendous number of webinars and training tutorials that you can go on at any time and kind of work through those as you're kind of working through and working with your data. And it's something that if you've looked at flow cytometry data, you can actually start picking this up pretty easily. So there was another question about the scanner and the Rhapsody Express, 2 different components. Joe, would you want to kind of discuss that a little bit about the different parts of the Rhapsody system?
Yeah, so as mentioned, there's 2 main components of the system, the scanner and the Express. The Express is essentially where the meat and potatoes of the assay occurs. It's where we isolate the single cells and harvest messenger RNA. It can be used in conjunction with the scanner. The scanner, I feel, has a lot of value in the fact that it provides quite a bit of upfront QC metrics that can evaluate the process as you're progressing through the preparation. It provides some QC metrics that essentially let you know whether or not the assay is progressing well enough to proceed through library preparation and ultimately go on to sequencing. One of the other key benefits that I like out of it is that it tells you precisely how many cells you actually captured. You may say, for example, load 20,000 cells, but in the worst case scenario, you only gather or you only capture, say, 10,000 cells. Well, now you know to set your sequencing depth recommendations or expectations for half of what you thought, so you can save money that way. But essentially, it provides a means to determine your viability, your cell capture rate, how many multiplets you may have, how many single cells are associated with a barcoded bead, things of that nature. So it's a really nice way to have some upfront confidence in what's ultimately going into the sequencing.
All right, very good. Thanks, Joe.
There's also a question regarding types of cells and variability of cell sizes that the cartridge can accommodate. Joe, can you make a comment on that?
Yeah, sure. Like any single cell assay, regardless of the platform, viability is absolutely critical. In terms of cell size, there is a point where, whether it be a droplet or a microwell, you eventually cannot accommodate larger cells. With the Rhapsody assay, the cartridge can easily accommodate cells of about 25 microns. After about 25 to 30 microns, you will start to see a small degree of the drop-off of capture efficiency. But typically, I'd say I recommend people for having the most or the maximum degree of capture efficiency, I recommend staying below 30 microns, preferentially below 25 microns.
Thank you very much for that, Joe. So there is a plethora of questions that we are really, really excited. We will make an effort offline to answer all of those questions and reach back to you with the answers. We've gone past 15 minutes over top of the hour. It's a long presentation. I really want to take this moment to thank you, Aaron, for engaging us so keenly, Joe, for all of your support, and all of you in the audience who have made this engagement very, very interesting and very, very informative, both for us and hopefully you guys got to learn a little bit of something that we think is very, very engaging. If you have any more questions, or if you want to have more information regarding the assets or this particular presentation, or you want to engage us in any way, please feel free to reach out to your local BD associate or come visit us on the website, and we'll be more than happy to accommodate all requests. With that, thank you, everybody, and have a wonderful day and have a wonderful week and stay safe. Thank you.